In this lecture, I will explain the mechanism of bacterial translation step by step. Let's begin. First, I will explain the formation of the translation initiation complex, AAA, in the 30S small ribosome. The start codon for translation is AUG, but the amino acid attached to the transfer RNA that pairs with this translation start AUG codon differs between eukaryotes and archaea, and bacteria and organelles. Additionally, AUG is not exclusively a start codon, and can also appear within the open reading frame. The transfer RNA that pairs with the translation start AUG codon is called Initiator transfer RNA, while the transfer RNA that pairs with other AUG codons is called Elongator transfer RNA. In eubacteria, chloroplasts, and mitochondria formal methionine is attached to the 3' end of the initiator transfer RNA. On the other hand, methionine is attached to the elongator transfer RNA. On the other hand, in eukaryotes and archaea, methionine, rather than formal methionine, is attached to the initiator transfer RNA, corresponding to the translation start AUG codon. Similarly, methionine is also attached to the elongator transfer RNA. The AUG codon used for translation initiation has the following characteristics compared to AUGs that appear within the open reading frame. In the translation initiation AUG codon, a Shine-Dalgarnosequence is present, 4 to 8 bases upstream of the AUG. The Shine-Dalgarnosequence refers to a sequence complementary to the 6 to 7 bases at the 3' end of the 16S ribosomal RNA in the 30S ribosome. In the case of E. coli, the sequence AGGAGGU corresponds to this. The anti-Shine-Dalgarnosequence, located near the 3' end of 16S ribosomal RNA, is thought to stabilize the attachment of mRNA to the ribosome by forming hydrogen bonds. On the other hand, such Shine-Dalgarnosequences are not found upstream of AUG codons that appear in the middle of open reading frames. Prior to the initiation of translation, it is necessary for the small subunit to be dissociated from the large subunit. Initiation factor 3 attaches to the area near the anti-Shine-Dalgarnosequence of the 16S RNA. The attachment of EF-3 to the small subunit facilitates the recruitment of mRNA. Additionally, through interactions between the Shine-Dalgarnosequence of the mRNA and the anti-Shine-Dalgarnosequence, the translation initiation AUG codon is positioned at the P site of the small subunit. Furthermore, initiation factor 1, which attaches near the A site, also promotes the binding of mRNA to the small subunit. In this state, the initiator transfer RNA with attached formal methionine forms a ternary complex with the EF-G GTP complex, allowing it to stably bind to the translation initiation codon. Once the setup of the translation initiation transfer RNA is completed, the large subunit is recruited onto the small subunit. When the large subunit associates with the small subunit, the GTP in the EF-G GTP complex changes to GDP, causing the EF-G GDP complex and EF-1 to dissociate. Shortly after, EF-3 also dissociates. Once this state is reached, the ribosome has completed its preparation for protein synthesis initiation, and can accept an aminoacyl-transfer RNA into the empty A site. Let's review the sequence of events using a different diagram. The translation start codon is positioned at the P site of the small subunit, where the initiator transfer RNA is recruited. Then, the large subunit is brought in. The EF-1, EF-2, and EF-3 used for setting up the translation initiation complex dissociate from the ribosome. From here, I will explain the elongation reaction of the nascent peptide chain. The initiator transfer RNA is in the P site, and to its right, aminoacyl-transfer RNAs randomly access the A site. The aminoacyl-transfer RNA is not transported to the A site alone, but rather in a complex with elongation factor TU-GTP. Please be sure that EF-G GTP is used for the setting up of formal methionine initiator transfer RNA at the P site initiation AUG codon. When an aminoacyl transfer RNA enters the A site, it is checked whether its anticodon forms stable hydrogen bonds with the codon. If a stable bond is formed, the GTP of the EF-G GTP complex that brought the aminoacyl transfer RNA is AAA, hydrolyzed to GDP. The energy from this hydrolysis causes a conformational change in EF-TU, and the EF-TU GDP complex is AAA, ejected from the ribosome. When EF-TU GDP dissociates from the aminoacyl transfer RNA, the formal methionine attached to the initiator transfer RNA is transferred to the amino acid, brought by the A site transfer RNA, forming a dipeptide. At this time, only the large subunit shifts by 3 base pairs towards the 3 direction. This amino acid transfer reaction occurs using the energy released when the aminoacyl bond is hydrolyzed. The catalyst for this reaction is the peptidyl transferase center located within the 23S ribosomal RNA in the large subunit. The EF-TU GTP complex is responsible for bringing aminoacyl transfer RNA to the A site. After delivering the amino acid and detaching from the ribosome, the EF-TU GDP complex is converted back to EF-TU GTP for reuse. This figure shows the ribosome in a state where the large subunit has shifted 3 bases in the 3 directions simultaneously with amino acid transfer. At this point, the E site, P site, and A site of the ribosome become hybrid sites where the spatial characteristics do not match between the small and large subunits. The initiator transfer RNA has already released its formal methionine, so its 3' end is now O and is located in the E site of the large subunit. However, since the small subunit has not moved, its anticodon stem remains in the P site. Similarly, for the transfer RNA carrying the dipeptide, its 3' end aminoacyl stem is in the P site of the large subunit, while its anticodon stem remains in the A site of small subunit. Next, the elongation factor EF-G GTP complex enters the now empty A site of the large subunit. When this GTP is hydrolyzed to GDP, the energy released causes the small subunit to shift 3 bases in the 3 direction, resolving the hybrid sites that had formed between the large and small subunits. Once the small subunit completes its movement, the transfer RNA that has released its amino acid is positioned in the canonical E site, and the transfer RNA with the attached dipeptide is in the canonical P site. The initiator transfer RNA in the E site will eventually be ejected from the ribosome. This figure illustrates a ribosome in the process of translation, with a growing peptide chain. In the left image, an aminoacyl transfer RNA and EF-TU GTP complex are entering the A site. Once the codon-anticodon pairing is confirmed to be correct, AAA, GTP hydrolysis occurs, and EF-TU GDP dissociates from the ribosome. Simultaneously, only the large subunit shifts 3 bases in the 3' direction, forming a hybrid site. Now empty A site of the large subunit is filled by an EF-G GTP complex. The energy from the hydrolysis of this GTP causes the 30S small subunit to shift in the 3' direction, resolving the hybrid site. As a result, the peptide chain is transferred, and the empty transfer RNA, now positioned in the regular E site, dissociates from the ribosome. for the peptide chain to elongate by one amino acid residue. A total of 2 GTP molecules are consumed from EF-G GTP and EF-G GTP. In addition to this, the energy stored in the aminoacyl bond is also consumed. The elongation rate of peptide chains is approximately 2 amino acid residues per second in eukaryotes, while in prokaryotes, it's much faster at about 20 amino acid residues per second. The error rate is low, with about 1 error per 10,000 amino acids added, regardless of whether it's a eukaryote or prokaryote. This translates to an accuracy of 99.99%. Next, I will explain the mechanism of translation termination. There are 3 stop codons, UAG, UAA, and UGA. However, there are no transfer RNAs in cells with anticodons that can form stable hydrogen bonds with these stop codons. Instead, there are protein factors called release factors that can bind to these stop codons. In bacteria, RF1 can recognize UAG and UAA, while RF2 can recognize UAA and UGA. On the other hand, in eukaryotes, a release factor called ERF1 can bind to all 3 types of stop codons. Although ERF1 is also a protein molecule, its 3-dimensional structure is known to be similar to that of transfer RNA. In contrast, the 3-dimensional structures of RF1, RF2 and eubacteria do not show similarity to transfer RNA. This suggests that, while ERF1 and bacterial release factors are proteins that perform the same function of translation termination, they are not considered to have evolutionary continuity. As translation progresses, a stop codon enters the A site. At this point, the P site contains a transfer RNA with an elongated nascent peptide chain attached. An appropriate release factor binds to the stop codon. The release factor then acts on the peptidyl transferase center, located in the 23S RNA of the large subunit, catalyzing the hydrolysis of the aminoacyl bond between the nascent peptide chain and the transfer RNA. The released nascent peptide chain exits through an exit tunnel in the large subunit of the ribosome. As a result, only the empty transfer RNA and the release factor remain in the ribosome. For the ribosome to initiate translation again, the empty transfer RNA and release factor must be removed, and the large and small subunits need to dissociate. This process involves release factor 3 and the ribosomal recycling factor. I have explained the mechanism of translation in bacteria. That's all. See you again soon.
If you have not found it yet, keep looking.